So welcome back, everyone. We're now going to move on to our next topic in this class, genome polymorphisms. And genome polymorphisms relate to the fact that once we start having entire genome sequences, we can start asking how these genome sequences differ from one another. So we would sometimes talk about having the human genome, but of course there isn't one human genome. Even in a single person, if you look at any particular region of chromosome, usually we have most of our sequence in the form of autosomal chromosomes. So we have paired chromosomes donated by our mothers and fathers. And those chromosomes aren't going to be exactly the same. If you look at any given little region of genome, every so often you're going to see a base that is flipped in one chromosome relative to the other. And those would be examples of genome polymorphisms. So usually subtle differences between the genomes of different members within a species. And of course, you'll see these polymorphisms in even a single person, but as you look at more and more people, you're going to see more and more polymorphisms. So when we start lining up lots of copies of a single genomic region across many individuals of a species, more and more of these variations will become apparent. This is a bit of an exaggeration of how frequently they are, but these are pretty frequent in the genome. Depending on the frequency threshold we set, we might see these every 100 or so bases. And that is what we mean by genome polymorphisms. So the study of genome polymorphisms goes back a fairly long way. Today we can look in a genome browser and we can see lots of information about known polymorphisms in the genome. So this is an image from the UCSC browser, just a few kilobases of sequence, and each of these tags here is shown in annotation in a track of genome polymorphism or genetic variation annotation corresponding to a single one of these bases where we know there are differences between the sequences of different copies of this particular region of the genome across people, sometimes between the homologous chromosomes of a given person. And there's a lot of interest in studying this kind of variation. So as I say, this has a long history. It's gone back a fair way. But this is becoming more and more a topic of study and more promising as a way of answering hard questions because so much data is becoming available now that it's gotten easier and easier to get this data. So one reason we are often interested in this is simply basic research. So genetic variations give us insight into ancestry and population history. So if we want to know how modern human population groups arose, so what was our history as a species, how did we develop into the different populations we see today, genetic variations are one of the important clues and really becoming the dominant source of information we would use to make those kinds of inferences. Much more often, this research is really driven by biomedical interests. So these variations allow us to look for correlations between genetic variation and phenotype. And very often by phenotype, we're talking about disease phenotype. So in particular, we can try to track down genes that may be implicated in a particular disease, or there may be targets for drugs for a particular disease, by looking for genetic variations in those genes that are correlated with whether or not you have that disease. And towards the end of this class, and much more in the next class, I'll be talking in some detail about how we actually do that kind of assignment. But for the moment, just be aware that this is primarily the sort of analysis we're using these for. And what I want to talk about before I get to that is much more what the data looks like. So what are genetic variations? How do we learn them? And what kinds of resources do we have for examining these? So there are some genetic variations that I will refer to as classic genetic variations. And by classic, I mean that these were variations people looked at really before the genomic era of biology. So before we really started sequencing DNA on any kind of large scale, the study of genetic variations actually predates the study of whole genomes by quite a bit. But there were some significant limitations and restrictions on the kind of variation you could look at. One of the really important kinds of variations people looked at is what's called a microsatellite variation. You'll sometimes see these referred to as short tandem repeat or STR variations, basically the same thing. And what an STR variation consists of is simply a small repeating pattern of sequence that is repeated different numbers of times in different copies or different people's copies of a particular region of genetic variation. So here, for example, we might have a repeat of the nucleotides GC, and different copies of this chromosome have different numbers of GCs in them. That would be what a microsatellite might look like. We would have a common sequence context, so identical sequence, non-repetitive sequence on either side of this, typically, but then we would have this repetitive region that is expanded or contracted to different degrees in different people or different copies of the chromosome. So these variations tend to have some important uses. They're still used, and I'll say a bit more about that later, but they tend to be nice kinds of markers, in part because they mutate very quickly. So these repetitive regions in a genome are problematic for the DNA replication machinery. It tends to make mistakes when it gets into these repetitive regions. So there's a high probability in copying one of these over the course of meiosis that you end up copying in an extra copy or a fewer copy, or one fewer copies, which is why these things tend to mutate quickly and tend to be good markers of evolution on the time scales of, let's say, human species or even shorter times. And if we have a variation like this, or maybe we have two different alleles and two different lengths that occur with high frequency, then we can, as we look across a population, we might observe that different people have different combinations of these alleles. So it might be that some people have the more common or major allele, and if they have two copies of that, we would refer to them as homozygous major. So homozygous major, that means that they have two copies of the more common allele, so one copy on each of the homologous chromosomes in that region. Someone can be homozygous minor, so minor refers to the less common version of the allele. And bear in mind, major or minor, either one could be longer or shorter. You may have more than two of these, but most commonly we're looking at things that have two possible variations. And someone can be heterozygous, or be a heterozygote in this region, have one of each. So one of the reasons people are interested in these is because they mutate quickly, and that makes them a good marker for relatively short time scales. When we're talking about genetic variation, generations, so let's say 100 generations is a short time scale. 100 million years is a long time scale. 10,000 years or so is kind of in between, but generally not a long time scale when you're talking about molecular evolution. So when I refer to short time scales here, I'm talking about short in terms of numbers of generations of people. But basically you can have these distributions of these, and one of the reasons they're useful is they mutate rapidly. The other reason they're useful is because this was something we could look at in the pre-genomic era. I'm showing you what it might look like if you sequence the DNA in the region of one of these, but these things were in widespread use before sequencing became a tool that people were routinely using. And the reason is because you don't need sequencing to type these. You don't need sequencing to figure out which version or which combination of alleles a given person has, as long as you know some basic facts about the microsatellite. So if you know the surrounding non-repetitive sequence, and you have some unique pieces of sequence on either side of these, and you know the lengths of the pieces in between there, so you know the lengths of the alleles, then you can actually come up with a simple test using pre-genomics of pre-sequencing technologies to figure out whether a given person is homozygous major, homozygous minor, or heterozygous. In particular, what you can do is apply a couple of tools we've seen previously in this class. First of all, PCR. So you generate primers on either side of your sequence. You copy out the region that contains the microsatellite. And then we need a technology for figuring out which version we have, and we can do that by trying to sort the sequences based on their lengths. So if we can figure out the length between the primers, we know which alleles we have. And we've seen a technology for that as well, polyacrylamide gel electrophoresis. So if we create our primers, polymerize out our sequences, you can run those out on a polyacrylamide gel, and the farther they run on the gel, the smaller the piece you copied. So if we have a couple of different lengths, then you could identify that someone who's homozygous for the shorter length would have a band that runs far on the gel. Someone homozygous for the longer length would have a band that would run less far. and a heterozygote would have 2 bands at the different positions. So it was relatively easy to type these kinds of things and figure out what variations we were seeing. I'll just briefly mention another kind of classic polymorphism, not as useful and maybe not as widely used, but another one that was useful in the pre-genomic era because it could be typed with standard tools of basic molecular biology, and that was something called a restriction fragment length polymorphism, or RFLP. So a restriction fragment length polymorphism is something that comes from restriction enzymes. And a restriction enzyme is simply an enzyme isolated from a bacterium that happens to identify and cut a specific pattern in DNA. So lots of these enzymes are known now, each of which will recognize a specific pattern. So if we have, let's say, a double-stranded piece of DNA, if you happen to have the pattern GAA-TTC, it's a genomic palindrome, which means it reads the same way on a complementary strand. There is a restriction enzyme called EcoR1 that will recognize exactly that sequence and cleave the strand in a particular way if it has exactly that sequence. But if someone happens to have a single base different in that sequence, so you don't have exactly that pattern in the EcoR1 restriction site, then EcoR1 will not cut that sequence. And that created another kind of polymorphism you could look for before we had genomic tools available. In particular, what we could do, as long as we had identified that one of these had occurred, and we could figure that out by trying different restriction enzymes, we could, just like with the microsatellites, come up with primers to the non-polymorphic pieces of sequence on either side of this, do a PCR reaction, copy out the piece of sequence within this region here, apply the restriction enzyme, and then we just need to know, did the thing we copied with our PCR get cut by the restriction enzyme or not? We can figure that out just as with a microsatellite by running it out on a polyacrylamide gel. And if we had a piece of sequence that happened to have the version of this that has a restriction site, you would end up with 2 shorter strands that would run far in the gel. So in particular, if someone were a homozygote for the version that has the correct restriction site, you will see 2 bands running far in the gel. If they were a homozygote for the version that is mutated, so it lacks the restriction site, then you will see a single band that runs less far in the gel because the thing you copied with PCR doesn't get cut. It's bigger and there's just one piece. And if you happen to have a heterozygote, you'll see 3 bands. So you'll have 1 band corresponding to the chromosome copy that wasn't cut, 2 bands corresponding to the one that was cut. So all of that is giving you a way of typing RFLPs and figuring out for any given person, are they a homozygote for the version that has the restriction site, the version that doesn't have it, or are they heterozygote? So those classic polymorphisms were useful in large part because they were easy to type. You didn't need to do any sequencing, you didn't need the pre-genomic era technologies. They also had some other advantages like the rapid mutation rate of the microsatellites, but they have some pretty serious disadvantages. One of them, probably the most important, is that they're not that common in the genome. For RFLPs, obviously it matters which restriction enzymes you're considering, how common they are in the genome, but more or less you can get some basic estimates of how common they are based on how often you see that particular restriction site, how often you'd expect to see mutations in it, and basically you're talking about megabase distances between these typically. Likewise, microsatellites, you're talking about, on average, millions of bases between these, so megabase distances. And that's useful for long-distance mapping, for localizing things to relatively distant points in the genome. It's not going to be useful for what we call fine-scale mapping, which is what you really want to do if you're trying to track down genetic variations in particular genes. These are actually still widely used in some contexts. So microsatellites, for example, are still widely used in the field of forensics. If you've ever seen, let's say, one of those crime victims shows where they will do a DNA test on a blood sample or whatever, isolated from a crime scene, and someone will hold up something that looks like a polyacrylamide gel, and they'll say that they've identified their suspect. very likely what they're showing you is actually a gel corresponding to one of these microsatellite typings. That still is quite important in those kinds of applications. But for research purposes, and more today for the medical purposes for which we're primarily studying variations, these have been largely displaced by other kinds of variations. In particular, that brings us to what I would refer to as the modern genetic variations. And modern really means the kinds of genetic variations people started looking at once we started getting whole genome sequences. By far the dominant kind of these, the most common kind, is what's referred to as a single nucleotide polymorphism, often abbreviated a SNP. So the exact definition of a SNP varies from definition to definition. Different people define it different ways. But more or less what it means is a single base of DNA that varies between different chromosome copies and that occurs in the less frequent version with at least some threshold frequency. So the threshold frequency can vary between different definitions. 1% would be a typical threshold frequency, but we might pick a higher one. And people also sometimes disagree on exactly what these refer to. Does it really have to be one base flipped to another base? Is a deletion or insertion of a single base something that counts here? Sometimes you'll see the term simple nucleotide polymorphisms instead of single nucleotide polymorphism to catch somewhat broader classes. But basically, we're looking at these small localized changes in the genome. And these are by far the most abundant kind of variation. So the most common kind of change you'll see between any one person's genome and any other is these SNPs. So the estimates of how frequent these are kind of a moving target. The more we sequence, and the more we sequence, especially in more isolated populations, the larger the estimate of their frequency becomes, and therefore the smaller the estimate of the average distance between them. But a rough order of roughly speaking, we can say once every 100 base pairs or so, there is a SNP. Again, it depends on your exact definition, exactly which populations you're referring to, but ballpark 1 every 100 base pairs. And that is pretty frequent in the genome and good enough to allow you to pin down specific genes or even specific relatively fine-scale locations in specific genes. So where do SNPs come from? Well, a lot of the SNP data initially started accumulating concurrently with the production of the large sequence databases. And the reason for this is because there are often reasons to sequence the same thing over and over. So if people were interested in studying a particular gene in the human genome, they might be sequencing that gene in lots of different people. And so a database like GenBank that is collecting these sequences would start collecting lots of examples of the sequences and be able to figure out when you were looking at regions that were consistent across your examples and when you were seeing variations between these. So the early compiling of these genetic variations, or the SNPs in particular, was largely something that kind of came almost for free from putting together these sequence databases. You could just look at when you had different examples of the same sequence isolated from different individuals and identify these variations and start collecting them into databases. So the NCBI, the same people who hosted GenBank, put together a data set called dbSNP that at least initially was collecting lots of these kinds of automatically generated variations, and then became a place people could systematically deposit variations however they get them. As sequencing whole genomes became a more common thing to do, the really first comprehensive libraries of whole genome SNP sets came about through this process. So by sequencing populations of people, at least initially when you sequenced a human being, for example, you wouldn't really sequence one person, you would throw in genetic sequence from multiple people. And by putting all of that together, we could see not just where those sequences lined up, but where there were subtle differences between them. And the first whole genome libraries of SNPs came, again, as a kind of lucky happenstance from the process of building genomes. More and more often, these are coming through systematic resequencing. So in many cases, we now want to know specifically about these genetic variations, and huge studies have been conducted to try to identify these variations. To take a genome we already have, let's say the human genome, and then start systematically looking at lots of different people. It's much easier to sequence a genome if you already have a very similar genome available to you. So you can resequence at a much lower coverage, align the reads from that low coverage genome against a reference genome, and more or less just look for what's different. And so it becomes possible to start resequencing on a relatively large scale. And there have been many projects now to do this. So a couple of examples of this have been the ENCODE project, the 1,000 Genomes project. I'll talk about some others of these later. But basically projects that for one reason or another are interested in looking at variations in sequences and gathering lots of different sequences. So that more and more often is how we're getting large numbers of genetic variations in our data sets. And this is, of course, something that is not just done with humans. People are interested in lots of organisms for lots of reasons, whether because they are important model laboratory organisms, they're important for agricultural reasons, they're important for other research reasons, and so forth. All right, so that basically is where SNPs come from. But if we want to do these kinds of studies, and if we want to use this information, we need some basic technologies for finding and working with SNPs. Classically, if you wanted to type your SNPs, you wanted to do a study of SNPs in a single population, you would more or less do this through PCR, very similarly to how I described the classic variation studies working. You could get PCR primers on either side of a base of interest and then more or less do something equivalent to sequencing a small region of interest or doing a simple test to figure out if there's a single base change from that region you copy out. You could do more subtle things like trying to sit your primers on top of your variations. That might work to give you a simple test. But basically PCR was the way you could do this when people were looking at small numbers of variations in small numbers of people. As this started to become something that people wanted to do on very large scales, we needed more high throughput technologies. And the first technology that really made this something that could be done in large sets of populations was something called a microarray. So microarrays I'll mention in other contexts. The SNPs are not actually the major purpose for which these are used, but they're worth knowing about for the SNP context. And what a microarray is, basically is a kind of slide, like a glass plate, that is covered with lots of little spots of DNA. So each spot will have a particular DNA sequence repeated many, many times, but different spots will have different sequences. It might be that this spot has the sequence AGC, TAGC, and this spot has the sequence AAA, TTT, or whatever you want. And a microarray will be covered with lots of these given spots. And a microarray can be used to detect whether a sample of DNA has a particular sequence. In particular, if we take a sample of DNA, we can attach fluorescent probes to that DNA. So we're fluorescently labeling the DNA, so we can more or less measure quantities of that. And then you can break up that DNA, break it into little pieces with all these fluorescent probes attached. It's known as fragmenting the DNA, just like in sequencing. And you can wash that over your microarray. And what you expect to happen is that DNA will stick to the spots if the DNA you're washing over the microarray is complementary to the sequence of the spot. Now, that's not going to be perfect. Some spots won't anneal nicely, maybe because the strand at the spot folds back on itself. Other spots may be cross-reactive. You may get things that don't exactly match that anneal to a spot. But more or less, you can kind of abstractly say that that DNA will attach to spots whose sequence is complementary to the DNA. And what this gives you is a test for the presence or absence of small pieces of DNA. So if you have set up your spots so that every piece of DNA you're interested in, every little sequence, has a spot complementary to it, then you will get fluorescently labeled DNA attaching to precisely those spots complementary to the things of interest to you. And then by shining a black light on this or shining the right frequency of light, you can get the spots to light up to show you which pieces of DNA were in your sample. And that was something we could turn into a way of typing which SNPs are present. So in particular, if we have some spots that correspond to some variants at a SNP site, other spots that correspond to a different variant, and then depending on which spots light up, we can figure out which variant a given person has. So this gave the really first fast, cheap technology. Cheap is always a relative term in laboratory context, but basically cheap technology where you could take a person and you could type their SNPs for a large number of SNPs relatively easily. So you could take a given person's DNA sample, you could run it over a microarray. Microarrays, at this point, cost on the order of a couple $100. You could run this over a microarray, you image the array, and then based on the spots that have lit up, you can figure out for hundreds of thousands, potentially even millions of SNPs, which variant that person has. And that really is what began to make it possible to start systematically typing large numbers of SNPs in large numbers of people. So it's true now, and it's been true for a number of years, that you can go to a catalog, you can order a pre-made human SNP microarray, or a mouse SNP microarray, or a dog SNP microarray. They're available for lots of different organisms that would have spots corresponding to the most common or most important genetic variations in that organism, and it made typing something that could really be done on a large scale, and started to make it possible to look at thousands of people in many populations and systematically study genetic variation. Today, it's largely true that resequencing is displacing these array technologies. It's not quite there yet as far as being cheaper and faster than microarrays, but it has some significant advantages. So today, resequencing is probably the dominant way we learn about genetic variations, but these SNP arrays are still around, they're still useful, even if they are kind of fading away as the technology for doing this. So just to show you, this is an example of what a real labeled microarray would look like. So here we see spots lit up in yellow here. There's some subtlety to different colors because usually you would have a case and a control, so two different samples, one labeled in red and one labeled in green. The detail we'll get to more often when we, well, later on when we look at another use of these in the context of measuring gene expression, but this is just to show you what an actual microarray looks like. So that is primarily how people are getting these SNP sequences today. Now, SNPs are not the only kind of variation of interest to us, and there are other variations that people are studying systematically in the human genome. And one of the other important classes that has really become an emerging area of study, really started taking off maybe 10 years ago, but is still being cataloged, is what's called structural variations. And structural variations are larger scale kinds of changes in the DNA that really to a large degree we didn't know were common in the human genome until we started getting lots of human genomes and could see how they might be different. So just like with SNPs, there are different definitions of exactly what we mean by a structural variation. But a typical definition would be a piece of DNA that is at least a kilobase, so at least 1,000 bases long, that appears in different numbers of copies or orientation in different copies of a chromosome. So the most common and well-studied of these are what are called copy number variations, or CNVs, which would consist of pieces of DNA that may be repeated, very much like a microsatellite, but much longer pieces of DNA. So in a microsatellite, we might have 2 or 3 bases of DNA repeated in different numbers of copies in different individuals. Here we might be talking about kilobases or more of DNA, a piece repeated, maybe not exactly, but more or less repeated in different numbers of copies in different people. A special case of that is an insertion or deletion or an indel variation, where there might be a piece of DNA that is simply found in some people and not found in others. We generally hope that it's not something critical if that happens, but we now know that some of these are relatively common. There are pieces of DNA that are found in a large fraction of people and not found in other people. You can also have inversions, large pieces of DNA, potentially megabases of DNA, that are simply flipped in some people relative to others. And again, this is something that we didn't really appreciate until relatively recently. And the reason we didn't appreciate them is until we had whole genomes and ways of systematically studying them, we had no way of knowing these were present or getting any real idea of how common they were. There may be other kinds of structural variations that we simply haven't discovered yet because we don't yet have the right technologies, or we're not asking the right questions informatically about the sequence data we have. So I wouldn't necessarily say that we've found all the kinds of structural variations we care about, but this is another thing people are looking at. By and large, structural variations are discovered through sequencing, and in particular through the pair-dense sequencing technologies that I mentioned previously, where we have fragments that have a known distance between them because they were sequenced by getting the endpoints of a larger piece of DNA. So we know the length of the DNA, we've sequenced both ends of that, and then we can get two fragments of DNA, or two reads, with a approximately known distance between them. And these kinds of pair-dense sequences are the major way we would figure out that there are structural variations in the genome. So, for example, let's imagine that there is a copy number variation, a CNV, between copies of the genome. So we might have one version that is in our reference genome, and maybe that has two copies of this big piece of DNA. So maybe this is a 10 kilobase piece of DNA. But it turns out that you are doing resequencing, and you happen to have done pair-dense sequencing on someone who has 3 copies of that 10 kilobase sequence in their DNA. The way you would figure this out is that you would get this pair-dense sequence. You would know a particular distance between the ends, and maybe you know that there is 50 kilobases between the ends of this, but then you map them back to the reference genome, and on the reference genome, you see that the ends are sitting a different distance apart. In particular, you can see that the ends, according to the reference genome, are only 40 kilobases apart. That would tell you that there is an extra 10 kilobases in the genome you just sequenced that is missing from the reference sequence. And then, if you knew that you were looking at a repetitive region of sequence here, you could figure out how many extra copies of that repeat were missing from your analysis, and figure out that there was a variation that had 3 copies instead of 2, let's say here. It's basically the same principle if we are talking about an insertion or a deletion. If we have a piece of DNA that's in our reference genome, we get a pair-dense sequence to someone who's missing that piece of DNA, then we will see that when we align the 2 ends of the pair-dense sequence to the reference genome, they will seem to have the wrong distance between them. They'll seem to be a different distance apart in the reference genome than the distance we actually know that strand has, and then we could figure out that either something is missing from our new genome or that something was inserted in there, and we could figure out the approximate length of it based on the discrepancy between the actual length between those paired ends and the one they seem to have when you map them to the reference genome. Finding inversions is a little trickier, so you could figure out that you have an inversion in part because the paired ends would be pointing in opposite directions in your actual sequence relative to what they seem to be, the directions they seem to be pointing in the reference sequence. You would also, in the case of an inversion, typically get a discrepancy in the distance between these because something that maybe is close to the 5' end of, or let's say close to the 3' end of this, would be moved further to the left in this version of the genome than this one. So you could also put together that evidence and figure out that you were looking at an inversion variation. But that basically is how we find these out. If you know a particular variation is there, you can test for that. You can type it using PCR. So if you know what you're looking for, you can get primers outside of these structural variation regions, and based on the lengths of the sequences between these, figure out how many copies you're looking at for a CNV or for an insertion or deletion. For an inversion, you could do other more clever kinds of things. For example, figuring out that you're getting a set of primers that will only prime if it's in one direction versus the other direction. And there are various ways you can go about this. More and more often, this is not something you would actually do. You would just run the paired-end sequencing and figure it out from your paired-end sequence. You don't actually type for known variations unless there's a particular one that's especially important to you. In some cases, we can also bring in the microarray kinds of technologies. So just as there were SNP arrays, you can also get copy number variation arrays. And this relies on the fact that these arrays are not just a binary measurement. So I had discussed with the SNPs before that you'll have these spots that light up when you have a complementary strand, don't light up when you don't have a complementary strand. But you get more than just a yes-no signal, you get some quantitation. So you can more or less figure out how many copies or how many doses of something are in your genome based on the relative degree to which a given spot lights up. So if you know how much a spot lights up, if you have one copy in your DNA sample, then something that lights up twice as much, you can figure out maybe has 2 copies. It's not quite true, but more or less that's the idea. The brighter it lights up, the more copies you have. And so you can come up with arrays that can give you at least approximate quantitation of copy number variations. So those are structural variations. As I say, these are something whose importance was not really appreciated until recently. It's worth noting that these are not nearly as common as SNPs in the genome. So rough estimate is that there's something on the order of 100,000 of these copy number variations in the human genome. So regions where you have a copy number change, or structural variation, I should say, not just copy number variation. So regions where you have a structural change involving pieces of more than a kilobase of DNA that are relatively common in the genome, on the order of 100,000 of these. Probably those numbers will go up as we sequence more people. But nonetheless, these are believed to account for a relatively large fraction of variant bases. So even though there aren't that many places where you have a megabase size inversion of the genome, each one of them may be accounting for 1 million bases of DNA that have some change in their relative context. So there is emerging evidence that these may be quite important for some phenotypes, especially disease phenotypes, and this is still an active area of research, but there are now some nice examples of some of these that are known to be correlated with various complex phenotypes. So just be aware this is another kind of variation, not as well studied as SNPs, but quite important. So just to give an idea of what these kinds of things look like on the genomic scale, here is a picture of a piece of genetic sequence taken from chromosome 19. What this is showing here is 200 kilobases of sequence. That's the genomic axis at the top. What we see here in these A's, C's, G's, and T's in red and blue is showing genotype SNPs. So these are SNPs that were looked at in one of these systematic studies of genetic variation, where someone has gone through various populations and tried to figure out which variations are found in particular people, and particular which combinations of variations are found in the same people on the same chromosome. That's known as genotyping. And there's more subtlety to that, but more or less that's the case. So lots of these have been studied in these systematic studies, and it's a little hard to make out here, but this blue line here that's a little broken is actually made up of little triangles, and those triangles each represent one SNP, so one known site of variation in the human genome. And at this level of resolution, they almost completely run on top of one another. You really can't make out the single SNPs because there are so many of them. So if we're looking at 200 kilobases of sequence, we would expect something on the order of 1,000 SNPs within that region. So that's a lot of variation. If we zoom out to a larger scale, this is looking at megabase sizes region of genome, we can start seeing some of these large structural variations. So this is showing various studies that were done to try to profile structural variation, and that we're identifying a large insertion-deletion region that seemed to be visible on this scale, so the order of a megabase of DNA inserted or deleted. It's worth noting that each of these different studies here found that there was something going on in this region, but they all identified slightly different pieces of DNA. That may be because there actually are slightly different variations on this, inserted or deleted in different people. It may be because there just wasn't enough precision in the technologies for looking at these things, but more or less that's how this kind of study would work. All right, so that basically is what these genetic variations look like. And there has been a lot of work now to systematically study these, to figure out what the patterns of genetic variations are across human beings, across different human populations, and more often to look at other organisms where we care about patterns of genetic variation for one reason or another. So the years for studying the human genome in particular. A lot of this got started with a study called PapMap study, which took off almost immediately after we had the first human reference genome to try to systematically profile genetic variations across human populations. There have been various others done by different groups, the Genographic Project, the Human Genome Diversity Project, the Population Reference Sample. Each of these has different kinds of advantages, looking at different kinds and numbers of variations, different sizes of populations. Some of them look at more people in a given population, some look at more different populations that tend to be useful for different questions. One really nice one that had produced quite a lot of data through systematic resequencing is the 1,000 Genomes Project. So a lot of the work in population genetics and human population genetics was really driven forward by this particular project with the goal of sequencing thousands of people spread across many human populations, so systematically resequencing to discover all of the genetic variations in those people. But basically, this is far from an exhaustive list. More and more is coming online all the time and looking at larger and larger scales. So more and more people putting together a sequence. And it's really quite realistic to think that we are going to have libraries of millions of people's sequences available for systematic study by the scientific community before too long. And this is really dramatically changing what we can do with these sequences and the kinds of questions we can ask. So this is already quite a dated list, and if you look at this a year from now or 2 years from now, it's going to be more and more dated and more as time goes on. But some basic statistics as of the time that I'm recording this lecture. If we look at one standard reference data set, so the dbSNP data set, as of October of 2014, there were more than 80 million distinct validated human variations in that data set, so more than 80 million sites where we know about a particular variation in the human genome. So obviously some of those are going to be relatively rare variations. We would expect something on the order of 30 million reasonably common variations. And systematic studies continue to be underway, as well as studies that are just happening for other reasons that are allowing us to discover more of these. There are hundreds of organisms available for which we have at least partial catalogs of variations now, and more coming online where we have really systematic whole genome libraries of millions or more of SNPs. So this is something that is becoming now a standard sort of resource available for looking at model organisms, looking at organisms that are important because of basic research reasons, and, for example, close relatives of human beings or other ones that are important in evolutionary studies. These kinds of libraries are now available for many important agricultural organisms. And as we'll see when we get into the next lecture, there are a lot of interesting questions you can ask when you have these kinds of data sets available, and a lot of kinds of studies we can do that become possible once we get this kind of information. And that brings us into very briefly the applications of this. So as I say, I'll be talking more later in the term in much more detail about these applications. Towards the end of this first half of the class, I'll be talking a bit about phylogenetics, the studies of evolutionary trajectories in populations. That's one of the major reasons we look at these. So for basic research reasons, we would like to understand where we came from as a species, and genetic variations are one of the ways we learn that. We more or less know that humanity began in sub-Saharan Africa. We were at one point a single population of people, and all the modern human populations spread out from that single small population and gradually spread over the world and made the modern diversity of the human species we see today. As that was happening, so as that history was going on, every lineage of human beings happened to be just randomly gathering mutations. Mutations happen all the time. Every generation will get a few new ones. And because of this, you get a kind of record of this population history. As long as you can figure out how to decode that, you can look at these variations, and by figuring out which groups of people have some common variations acquired over the course of our history, we can figure out how those people are related to one another, how far back their ancestry traces, and through modern computational methods, get really detailed models of how these histories evolved, where different populations groups came from, how they arose, how they split apart, how they joined together. This is really an active area of research, how exactly to do this, but it's really become the dominant way that we are learning about our history as a species. The other major reason, and the dominant reason why people are actually putting a lot of effort into these studies, though, is medicine. So largely this is talking about human medicine. Again, in some cases people are asking similar sorts of questions for agricultural reasons, so similar kinds of techniques apply, but just talking about human medicine. The major reason we look at these variations is because we're interested in what they can tell us about phenotype, especially disease phenotype. In some cases, a variation directly affects protein function. So an example of this would be Huntington's disease. Huntington's disease is a genetically controlled illness. It's a very serious degenerative neurological illness. And it happens to be controlled by a particular kind of short tandem repeat, essentially a microsatellite variation, in which we have a triplet CAG in the coding region of a protein called the Huntington protein. CAG happens to be the codon, or codon for glutamate amino acid. And so the more copies of this you have, the more glutamates you have. And it turns out that having a lot of glutamates is a bad thing. If you have lots of glutamates, you get aggregation of the protein, so these glutamate regions kind of stick to one another. It causes this Huntington illness. It's a very serious illness. And if you have a large number of copies, you can say with certainty, if you live long enough, you're going to get this illness. If you have, let's say, 10 copies of this repeat on each chromosome, you're never going to get Huntington's disease. If you have 40 copies on either of your chromosomes, if you live long enough, you will get Huntington's disease. If you've got 100 copies on either of your chromosomes, you will get Huntington's disease at a fairly early age. So there is a direct cause and effect relationship between these. A single base change can even cause disease directly. So the classic example of that is sickle cell anemia. So in sickle cell anemia, a single nucleotide in a codon changes a GAG to a GTG, which changes a codon for glutamine into a codon for valine. That produces an important change in the protein, creating a little hydrophobic patch on these proteins that allows the proteins themselves to aggregate and to produce what's called the sickle cell phenotype, in which red blood cells under certain conditions will start having their hemoglobin form these polymers of hemoglobin, distorts the red blood cells, you don't get proper blood flow. Again, it's a serious and debilitating illness. Variations can affect function in many other ways, though, sometimes much more subtle ways. So a polymorphism can affect splicing. If it happens to sit in an important splicing signal, you can get disease that way. You can get polymorphisms that affect expression, so activities of genes. So if you have a variation in a site that happens to be a transcription factor binding site, so a site that controls how the cell regulates, how active a particular gene is, that can, for example, change the level of activity of that gene, and that can produce phenotypes, both subtle and extreme, can change where a protein localizes. Lots of other things can occur. So there are many ways polymorphisms can affect phenotype. For the most part, though, when we look at these polymorphisms, we're not looking specifically for the polymorphisms that affect phenotype. We're often just trying to figure out approximately where in the genotype these polymorphisms occur by looking for statistical association, effectively correlation between a polymorphism and a phenotype of interest. So, for example, it might be that there is a polymorphism that causes a rare disease that sits in a particular region of a genome, but it's simply too rare to be able to identify that by itself. We're not going to be able to find that a very rare variant is associated with a polymorphism unless we already know more or less where to look. However, it might be that there is a much more common variation that is in statistical association with that variant, so that occurs more often than chance in genomes that have the rare disease-causing variant, maybe because it happens that the rare variant appeared in an ancestral lineage of people where this common variant was common. So because of this, what we usually will try to do when we're trying to study relationship between genotype and phenotype is find these indirect correlations. Find a correlation between disease and the common variant that happens because the common variant is correlated with the rare variant, and the rare variant is the thing that actually causes the disease. And we'll be seeing much more depth in the next class about how exactly we go about asking that kind of question. Where a lot of this is going, and a lot of the reason many people are interested in this, is that there's a widespread belief that we will be able to dramatically change the practice of medicine through what is called personalized medicine. And the basic idea behind this is that we want to change the paradigm of how medicine is conducted. At least at a high level, a certain level of abstraction, we can say that medicine has a particular practice. If you are ill, you go to a doctor, the doctor observes your symptoms, and then based on the symptoms, the doctor makes a diagnosis. Then, based on the diagnosis, the doctor will prescribe a treatment, and that's more or less how medicine works. At least in principle, though, your genetics, or any person's genetics, are giving the doctor a lot of information that might be useful in making these kinds of analyses. So if we know, for example, that you have a particular set of genetic variations, and we have lots of other people's genetic variations, and we know their medical histories, then in principle we could identify that maybe you have genetic variations that suggest that you are at very low risk for common causes of these symptoms, but at much higher risk for rarer causes of these symptoms. If that's the case, then the doctor in principle should be able to figure out, based on looking at not just your symptoms, but also the symptoms and genomes and diagnoses of lots of other people and your own genome, that maybe a less obvious diagnosis is the right one for you, and then come up with a treatment that is tailored to your particular genome, that maybe will take account of the fact that people with your genome respond more strongly to certain drugs than the average person and less strongly to other drugs. So both diagnosis and treatment in principle can be influenced by your genome. A lot of us working in this field would say that this is something that is going to change the practice of medicine, and I know many people who take this class are pre-meds, you're interested in becoming doctors one day. I would say that this is something you are going to have to know about. You are going to need to know a lot more about genetic variation and how it influences this disease than is the standard today if you want to be a practicing doctor 10 years from now or 20 years from now or so forth. Some of this is already making itself a big part of treatment practice. So in some special cases like cancer treatment, for example, this kind of personalized medicine is already really a reality. People are taking into account quite a bit the specific genetics of a particular patient's cancer in figuring out the right diagnosis and the right treatment. But this is very likely to become a much bigger practice, a much bigger part of medical practice in many different contexts. So I put that out there as something to be aware of for the future, why we care about this. And again, in the next class, we'll get much more into the mathematical and statistical work that goes into discovering these kinds of important genetic variations. So any questions about any of this before we break? Okay, then I guess we will stop.